Hello everybody, today is our Chordates lecture, our last phylum in the class this semester. This group is the group that we are in, so we like it very much. There is also a lot of fossil evidence of chordates because the internal calcified skeleton makes really good fossils, and so we have a fairly easy way of learning about the evolutionary history and ancient species in these groups. So we want to start with the basic chordate characteristics at the beginning, and then we're going to talk about the different groups and some of the characteristics that those different groups have. We have, for chordates, they are in the bilateria, so they're bilaterally symmetrical. They are triploblastic. And when we talked about development before, we have protostomes and we have deuterostomes, and we looked at one deuterostome phylum already, the echinoderms. Chordates are our second deuterostome phylum. They have a complete gut, and they have a eucoelom. So those are our basic body plan characteristics. And then we're going to look at the characteristics that are unique to chordates. The diversity of chordates is really quite fascinating. There's a lot of fish-like forms, but when we look at the most basic body of chordates, we're going to find that we have these two kinds of organisms here, the tunicates and lancelets. We'll talk about them in more detail in a minute, but these we're going to categorize as invertebrate chordates. And we usually use the term invertebrates to mean things like snails and worms, things without an internal skeleton. And invertebrates are distinct from vertebrates because vertebrates have vertebrae. So even though we have these taxa inside the chordates, they don't have vertebrae. However, they do have what we'll talk about in a minute, which is the 4 chordate characteristics. So we define them as being chordates, even though they don't have vertebrae. And the rest of the chordates that have vertebrae, we call them vertebrates. Right? So we have the invertebrate chordates, we have all kinds of other invertebrates, and we have the vertebrate chordates. So within the vertebrate chordates, we have these two taxa, hagfishes and lampreys. They're fish-like, but primitive in body form. We'll look at them in more detail. And then the next major evolutionary change we see is having these jaws and paired appendages. So when we talk about jaws, we don't just mean that they have a mouth. We mean that they have a top and a bottom jaw that meet together with bitey teeth in them that is quite handy for taking in food. So the new structure that makes these organisms have a jaw is really the lower jawbone, right? So when we have a skull, you see that this part is separate and it comes off, it's a separate bone. And so we have the formation of this jaw. And because of that, we have a monophyletic grouping of all the organisms that have that jaw, and we call them gnathostomes. Jawed mouth, gnath, GN. And what goes along with that is the paired appendages. Now, we talked about paired appendages in the arthropods, but chordates also have paired appendages, but we only have 2 pairs. We have our front legs, our pair of arms, and then we have our back legs, our hind limbs, or legs in humans, and that is a pair of pelvic appendages. So when we start to see these fish-like chordates, they're going to have fins, pairs of fins, the pectoral fins and the pelvic fins. And we'll see as we go through the evolutionary history of vertebrates how those fins evolved into limbs, right? So in order to live on land, just like when we had plants making a transition to land, they had to have some changes in their body to adapt from the aquatic environment to the terrestrial environment. So we have to have a change in structural support because you have to hold yourself up against the force of gravity on land. You have to have a way to waterproof the outside of the body, yet still be able to do gas exchange. So we saw that in plants. And then you have to have some way of doing reproduction, right? Because if the sperm is going to swim to the egg, then that has to happen in water. So we need to have a way that sperm and egg can get together, and then that early developing embryo can be protected in some kind of watery environment. And so we'll see that the next big evolutionary changes in our chordates were the things that allow them to move onto land. So the first things that evolved were the lungs, which actually evolved in some fish-like chordates that didn't actually live on land, but they sort of had the ability to breathe air when they were close to the surface. So we're going to look at the first step of becoming terrestrial as having those 4 limbs, right? So limbs instead of fins. And this defines a monophyletic grouping, right? And we're going to use, instead of 4 limbs, we're going to say tetrapods. So this monophyletic grouping of these land-going chordates is the tetrapods. Tetrapods, tetra means 4 legs, right? So that was the ability to withstand the force of gravity on land, move around on land, our limbs. Our lungs is going to allow organisms to breathe air on land. We also needed some better waterproofing. And so we'll see that another characteristic that occurs here with the amniotic egg is scales. So scales are a modification of the skin. It's reinforcing the surface of the skin with a protein called keratin that's unique to reptiles, birds, and mammals that is going to make a tough outer waterproofing surface. Now, the last characteristic on here is that amniotic egg. So even though we have the amphibians, which are shown on this group here with the frogs, salamanders, and secalians, amphibians are going around on land. They don't have a waterproof skin. In fact, their skin is thin and moist. So they're on land, but they're not fabulous at being on land. They have to stay pretty much in moist areas, and they have to do their reproduction in water. So they're going to have to find a pond or a lake or stream in order to lay their eggs, because sperm and egg are going to come together in the water. So amphibians are like our early terrestrial plants, mosses and ferns, where they still had to do reproduction in the water. So they're called amphibians because they have these 2 modes of life, on land and also in the water. The amniotic egg, like the chicken egg that we saw in the development lab, is going to have a membrane called the amnion. So the amnion is actually a membrane that surrounds the embryo. Inside of the membrane is a watery environment. So the embryo is going to be maintained in a moist environment inside that amnion. And then on the outside of the amnion, there is a shell that's produced by the body of the mother as she's laying those eggs. And it's going to allow the egg to be in a place where the environment can be dry and the embryo inside of it is still going to survive. So just like a seed, the egg is going to have some, all the nutrients and water and minerals and other molecules that the developing embryo needs to grow. There's going to be able to get oxygen inside of it through diffusion, through the shell of the egg. And so the embryo is then going to develop until it's ready to hatch and live on its own. So with the evolutionary invention of the amniotic egg, with scales to provide waterproofing to the body, we have the reptiles, which includes the birds and mammals. So fully terrestrial, 100%, all body functions are taking place in that terrestrial environment. And then in the mammals, we have an additional adaptation of not laying the egg outside the body, but keeping that amnion inside the body, connecting it to the body of the mother with a placenta. So we talk about mammals as being, as having a placenta or having development inside the body of the mother or the uterus. All right, so this is our brief survey of the evolutionary history and major characteristics of the chordates. And we're going to look at the basic body of a chordate. What are the characteristics that it needs to have in order for us to call it a chordate? All right. So all of the taxa that we showed in that tree diagram basically start out life as an early embryo that goes through development and has these 4 characteristics that it develops. Now, some taxa then lose these characteristics, but we can see them in the embryo or the larva. All right, so this little animal over here is an example of a lancelet or an amphioxus, and its body is basically the body of this early basic chordate. So, but I'm also going to draw a cartoon and show what these characters are. So we have the basic body. I'm going to emphasize it a little bit more than what we can see on our little tiny lancelet. All right. So here we have the mouth. We have a complete digestive tract. So we have mouth and we have anus. And the bodies of chordates are in general fish-like in that they're laterally compressed. And the way that they're going to do a body movement, locomotion, is swimming. So that's going to be a back and forth wiggling of the body. And the reason that they can do this back and forth wiggling is that they have a structural support called the notochord, and it is a dorsal longitudinal flexible rod. It's made of cartilage. So it's stiff compared to the rest of the body. So it allows the muscles that are fastened to it to flex, and it causes the body to sort of wiggle back and forth. And the notochord is going to be sort of this second line I've drawn along the body of the animal. So muscles are going to attach to it. It's going to be used as structural support and locomotion. Dorsal to the notochord is the dorsal hollow nerve tube or nerve cord. Right, sort of hollow and tube are kind of redundant. Now it's hollow, it's dorsal because it's on the backside. The anterior end of it is going to be the brain. And then it's hollow because during development, remember, we saw that those neural ridges rise up and merge together across the top. So we have this little circular space that is going to end up becoming that dorsal hollow nerve cord, so our spinal cord. And that is going to be indicated by this first line. All right, so it's closest to the dorsal side. The notochord is actually underneath it. In order to do this locomotion, it's going to be helped by having a tail. And so that's going to be a flattened, thin ending of the body so that as the body wiggles back and forth, it gives a more power to the swimming motion. And this tail isn't just anywhere, it's after the anus. Right? I don't know of any organism with a pre-anal tail, but we have a post-anal tail. It comes after the anus. Here is the anus. Here is the post-anal tail, or PAT. So, and notice that it is laterally compressed. When we look at a dolphin or a whale, we see that the flukes, the swimming motion that they're making, is going to be an up and down motion. But all of these primitive fishes, it's going to be a side to side motion. All right, so the combination of the postanal tail, the notochord, and the muscles are going to result in swimming, so structural support and locomotion. Now, the pharyngeal gill slits are a very interesting structure, and they're used for several things. So we have a requirement for this organism, it's a marine organism, to do respiration. So it's going to have to get oxygen in across a respiratory structure. And we think about the main structure that aquatic and marine organisms have in order to do respiration is gills, right? So we have these pharyngeal gill slits, and indeed, they function as gills. And what's going to happen is that water is going to come into the mouth. It's going to go into this region, right? So the anterior region of the digestive tract in us is called a pharynx, right? So our pharynx is a little tube that gets food into the esophagus. So these primitive chordates have a pharynx and it has slits in it, right? So we see that the mouth, the water is going to come into the mouth, and the slits are important because the water is going to go out them and into this space that's inside the sort of trunk region, and then out this other structure over here. So it has an anus for the waste products of digestion, and it has this structure which is called an atriopore, which is where that excess water goes through. So imagine you're taking in a bunch of water, it's going over your gills, it has to go somewhere. This is where it goes out. Now the gills also have a function of being used for filter feeding. And so food particles are also going to go in the mouth, and they're going to be kept inside. They're not going to go through the gill slits. They're going to go inside the pharynx, and then the food is going to go down the digestive tract, and the waste products are going to go out the anus. So the pharyngeal gill slits are both for respiration and for feeding, right? If we try and breathe and eat at the same time, we choke, but our primitive chordates breathe and eat at the same time without choking, right? So our pharyngeal gill slits are gill slits, and they're named for that because they are in the pharynx, all right? This one is a little hard to spell. So the pharyngeal gill slits are there, all right? So when we look at our basic body of our most primitive chordates, the cephalochordates, right? Cephala means head, so it's a headed chordate. We see in this organism. So this is called Amphioxus. They're also called lancelets or sand lancelets. All right, cephalochordates. It has a few of these little tentacles around its mouth for feeding. It has the atriopore here. Water and food are going to come in the mouth. Water is going to come out the atriopore. Food's going to go into the digestive tract and out to the anus. So the notochord and dorsal hollow nerve cord are a little hard to see and distinguish from one another in this figure, but they are on the dorsal side of the body. Now the other thing that's on here is myomeres. And myomeres are muscle blocks. And myomeres are repeated muscle blocks. And if we look at the body of our little sand lancelet, which when it's not under the microscope, it looks very unremarkable. The one thing that we may be able to notice is it has these little chevron shapes or little V shapes going down the side of its body. So if we're looking at the external anatomy, it doesn't really have a nice big eye for us to see. It doesn't have gill slits on the outside. They're very tiny. They're only a few centimeters long. They never get bigger than that. They're kind of pinkish, very light pink. Right, so this is our picture of our little sand lancelet here. And if you sort of squint at it and you sort of turn it a little bit in the light, then you can see these myomeres or these repeating muscle blocks, right? Now, other kinds of fish have them. And so when we are cooking a fish and we're going to look at the properties of the fish to see if it's done, right? We're actually going to see kind of these stripes, or the muscle tissue of the fish is going to twist apart. Like if we stick a fork in it to see if it's done and we twist, then those different muscle blocks, they're going to pop apart from one another, right? So each of those separate little strips of muscle are the myomeres, and myomeres are a reflection of segmented body plan, right? So we have these multiple repeating units represented in those muscle blocks in fishes, right? So our next group of invertebrate chordates are the urochordates. And we have these organisms as larvae have the four chordate characteristics. But as adults, they lose the tail and they lose the notochord. They are relatively complex organisms, however, but basically they've taken the organism that I drew on that first slide and you fold it in half and turn it over, right? So what we have is we have this oral siphon. This is the mouth. This is where the water and the food is going to flow. We have this branchial basket, which is the pharyngeal gill slits. Then water is going to move out through this atrial opening. And then we have food moving into the gut, right? This tunic, and the way they get the name tunicates is that they have this sort of skin-like structure over the outside. So when we're looking to find, see if an organism that we're looking at is a tunicate, the most obvious feature is going to be these two siphon openings, and they're going to be a little bit offset from one another. They're about the same size in diameter. And when we look at the body of these organisms, they can be opaque, but they can also be translucent. So they may look like a jellyfish-like animal, and these are some of our closest relatives, people. All right, so they're very weird, yet they have these important chordate characteristics. So we have a variety of different organisms that we're going to name that go in this category. I usually say tunicates. They may also be called salps, larvaceans, sea squirts is the common name for them, because if you pick one up and it's not in the water, it'll sort of tense its muscles up and close its siphons. If you squeeze it a little, sometimes it'll shoot water in a fountain out the top. So you can use them to squirt water at people like little squirt guns, which if we were in person in lab, I usually would squirt one at a person. So you're getting away from having that happen because we're on Zoom class this semester. So here's a picture of what they may look like. These benthic ones look pretty much like our basic diagram. We can see that there's many individuals. We can see these two openings, these two siphon openings when we look at all these different individuals, and that otherwise their body is just kind of a U shape, right? So we are going to look for that when we're looking at benthic ones, that is ones fastened down onto the bottom. And they often form a colony. So we're not so often we're going to see just one, but we're going to see many together, and they can be brightly colored. There's also pelagic, or ones that are up in the water, and they may form long chains like this, but each individual one here is kind of this cylinder shape with sort of blob at one end with maybe some dark stuff in it. They don't look like anything in particular. And I always struggle when I look at them to say, oh, that's a tunicate, clearly. The salps, basically, they're going to look like a gelatinous zooplankton, which is what they are. They're translucent, typically. You can't really see any particular structures. So you might think it's a jellyfish, but it does not have obvious radial symmetry. It doesn't have obvious bilateral symmetry either. But you want to look at some kind of clear, blobby looking, obviously marine organism. Look to see if it has radial symmetry, it has tentacles hanging down, it's a jellyfish. If you're looking at this thing and you have no idea what it is, it's just a blobby gelatinous thing, it's a tunicate. All right, so Eurochordates, they may not look like anything in particular. We do have some benthic ones that, instead of having a nice bright color on the outside, are more camouflaged, and they're more sort of leathery and warty looking. All right, so those are often hard to identify, but we're going to look for those two offset siphons at the top. All right, so now we're going to shift gears and look at our vertebrate chordates. So vertebrae are going to be structure that goes next to the notochord. So this organism, hagfish, has very few obvious characteristics. It's going to have tiny little holes on the outside of its anterior region. You can't even see them in this picture. It has tiny little squinty eyes. It has tiny little, excuse me. Some little tentacles around its mouth. Its body is smooth, though it's not segmented. It does have an obvious postanal tail. And then when we look inside of it, right, if we're gonna see the notochord, right, the vertebrae are just these little rods that sort of sit on top of the notochord, right? So the vertebrae don't start out looking as complex as the ones that are in our skeleton, but they are a series of repeating segmented structures. And they are going to be in our hagfish. So we have a vertebrate. And they're not reinforced with calcium, right? They're just cartilage. And they're just going to sit over the notochord. So the only thing that we're going to be able to notice about our hagfish is that it doesn't have any particular features except that it's not segmented. It does not have paired fins. So we do have eels that are in the bony fishes, but if we look at our eel closely, we'll see that it has the little pelvic pectoral fins. It has a dorsal fin. And we'll see some other structures that our bony fishes have. So just because it's long and thin doesn't mean it's an eel. It could be a hagfish. It could also be a lamprey. So hagfishes are hard to identify just because they don't look like much, but we have some examples in lab that'll help you see more of their, recognize their characteristics. All right, and their group name is Mixini. The lampreys are the other group of jawless chordates, so we're not to the gnathostomes yet. And this picture is showing you what the front of its mouth looks like. So it's hard to imagine what a mouth without a lower jaw is going to look like. It's basically just an empty circle, right? It can't close the front of its face, right? It just has an open end to its face, like a vacuum cleaner tube. Although this one has some raspy looking, scary looking teeth. So I think this thing has been made into some kind of scary monster in a science fiction or horror movie, because with those teeth and those face are coming at you, that's kind of scary looking. So it is a parasite. It does fasten itself onto a prey, other fish usually, and it sort of rasps a hole in the side of the skin and it sucks the blood and body tissues out. So not only do they look scary, they are scary. So when we look at the a mid-sagittal section, we take a longitudinal section through the head of the front of the lamprey, we can see that it has its brain, tiny brain, and the spinal cord, right, that are extending down the length of it. The notochord is here, and it's much thicker, right? We can't see in this picture because they aren't really called out, those little repeating units of the vertebrae. So they're there, we just can't see them. By the time we get to the hagfish and lamprey, if we start looking at the internal body organs, we see very characteristic chordate body organs. Now, the only thing that's different among most of the vertebrates is how many chambers the heart has. And otherwise, they all have the same basic nervous system. Terrestrial ones have lungs for respiration, while aquatic ones have gills. We have a liver. We have complete digestive tract. We have separate sexes in order to do reproduction. Anterior brain and a variety of sense organs that we may find on the body. And that we also have, what did I leave out? Oh, the circulatory system, we have the heart and blood vessels, so a closed circulatory system. All right, so all the fish-like chordates, basically everything that we've talked about so far, have a two-chambered heart. When we get to the amphibians and reptiles, we have a three-chambered heart, and birds and mammals have a four-chambered heart. So the other thing that's interesting about the lamprey is that they have a larva. So when they're juveniles, they're little baby lampreys, they look almost identical to Amphioxus. They look like those a little tiny, perfect little chordate, right? So let's see if I have. When we look at our lamprey from the outside, and we don't have a good, I don't have a good picture on this slide for you, but in the lab materials, there's a nice video that shows some preserved specimens. You can see what the front of its head looks like. The back end of its body looks more or less like a hagfish, but the front end of its body has a much larger and more obvious mouth. It still does not have any dorsal fins or pectoral fins, but its eye is much larger and obvious, and it has bigger gill openings that you can see. So if it looks like it actually has a face, it's a long and skinny thing that's not an eel, it's a lamprey. It's a long and skinny thing that's not an eel. It doesn't really have a face. It's a hagfish, right? Partly they're called hagfish because hag is another name for witch, right? So they're ugly. They're not attractive. All right. So when we make the transition between the lampreys and hagfish and the rest of our chordates, we make the transition between not having a lower jaw and having a lower jaw. So we're gonna build our gnathostomes now. So we see that if we're going to get to a place where we have this lower jaw bone, this that allows us to close our mouth, where does it come from? So as vertebrate evolution carries on, the internal body elements of the skeleton, which are reinforced cartilage. So this cartilage starts to get laid down into little structures sort of scattered throughout the body, in addition to around the notochord. And one of the places where these little skeletal rods appear is along the sides of those pharyngeal gill slits. As the body gets larger, in order to support them, make sure they stay open. All right, so these gill slits have these little skeletal rods associated with them. The way that the lower jaw is formed in evolution is by a gradual fusing together and migration forward in the face of the skeletal rods that go around the third gill slit, right? So we see that these red and these green bars, as they move towards the front of the face, they get larger, they develop a joint between them, and the upper ones fuse to the skull. So we have a strengthened and reinforced of those skeletal elements, and then we have a hinge that allows them to open and close. So it seems pretty obvious that this is an advantage to be able to bite food rather than trying to just suck it up, all right? So that this is a major characteristic that led to the success of all the other vertebrates. All right, so now looking at our gnathostomes, we're going to skip a couple groups, but the most common and obvious organisms in our environment, in the marine environment, are going to be of chordates, are the chondrichthyes or cartilaginous fishes, the sharks and the rays. All right, so sharks and rays range in size from adorable little tiny guys to enormous things like this whale shark. They may be filter feeders, and they may be scary predators. And so when we look at the diversity of sharks and rays, they're a pretty successful and very old group. And so they've been around through the evolution of marine organisms for a very, very long time. The two characteristics that we want to look at on their bodies that makes them distinct from the bony fishes are going to be and do a very good job of drawing this. But their mouth actually opens on the bottom of their face, right? So if we look at Mr. Big Tooth down here, his mouth is not right towards the very tip of its nose, it's more underneath his face, right? So especially when we look at our ray here, its mouth is definitely on the lower side of its body. We see that instead of having a little round opening, we have these large gill slits. We have our two pairs of fins, paired fins. And then we have a tail that has a longer part on the top than it does on the bottom, and that's called a heterocircle tail. And so we're going to be able to tell the difference between a shark and a bony fish because we can see this heterocircle tail, all right? We can see the gill slits and the location of the mouth, right? Now these paired fins, that comes along with the lower jaw, right? So we see the lower jaw, we see in our naphostomes, and we have these two pairs of fins. The pectoral fins, right? If you're doing pec activities, then lifting, doing bench in the gym, you're working the pectoral muscles. These are called the pectoral fins. And then our lower half of our body, we have our pelvis or our pelvic bones. These fins are called the pelvic fins in fish. So two pairs of fins. All right, Actinopterygii. This is one of the funnest names in this group to say. Actinopterygii. You got those two eyes on the end, and you have to do an EI in order to say it. Actinopterygii, the bony fishes. So the vast majority of fishes, that fish-like organisms that we see, are bony fishes. They range from tiny to gigantic. They live in every freshwater, marine, semi-aquatic habitat on the planet, the most diverse group within the chordates, right? So many, many more species of bony fishes than any other taxon. Ranging from our common goldfish that we might see with our basic body of a bony fish. So I'm going to draw right on it here to emphasize its tail, right? It has a little dorsal fin. It has its little two pairs of fins here, the pelvic and the pectoral. Its mouth is right at the very tip of its base. And then it has this structure. We see, instead of having a series of gill slits along the side, all of them are covered by this one structure. So that structure is called the operculum. And we see it's kind of like a half moon on the side of its face. So if we see that half moon on the side of the face, it's not really that obvious in any of these other fishes, but we can see it here on this giant catfish. You can see it here on this salmon. That we know it's a bony fish and not any of those other kinds. We see this dorsal fin. That one is going to tell us it's not a hagfish or a lamprey. And also the other paired fins. And a much larger and more elaborate tail in most cases, not just a basic little postingal tail. The other characteristic that the bony fishes have is their body's covered with scales. Now, I forgot to say, in the chondrichthyes, their skin is very rough. And so there are these little teeth-like or skeletal elements that are a unique type of scale that is on the skin, the body surface of the sharks and rays. The bony fishes are covered in scales that are different in structure than the scales that we find on terrestrial chordates like lizards. Now, our first group within the tetrapods is the amphibians. So when we go from strictly aquatic organisms that are fish-like, remember that we have to have our limbs. We have 4 of them. We have lungs. And those are the 2 characteristics that our amphibians have that allow them to live on land. Now, they don't have the amniotic egg. They have a fish-like or gelatinous egg. I'm not going to talk too much about amphibians. I find that most students are generally familiar with the idea of what an amphibian is. So frogs, toads, salamanders, and newts. Newts are actually a sort of special kind of salamander. And we either see that they have the 4 legs, the big jumping back legs, or their body plan, like the salamander, is a little more lizard-like. But we're going to be able to tell that they have, tell them apart from lizards in a couple of ways. First of all, the skin is smooth. They don't have any scales. I mean, they don't have fish-like scales. They don't have lizard-like scales either. They're going to support their very rudimentary lungs by also doing respiration with their skin. And they have a 3-chambered heart. They have, now most tetrapods have, we have 5 fingers and toes. Most tetrapods do, except amphibians. And so they only have 4 toes on their front limbs. All right, so a frog can't give you a high 5, it can only give you a high 4. That's it. It's just got 4 little toes. And the tips of the toes, you can't see it too well, but the tips of the toes don't have any claws on them. They don't have any nails like we do on the tips of our fingers. They're sort of just blunt. All right, and they're going to have to do their reproduction in water, so they're not completely terrestrial. We're going to do a frog dissection, and we're going to see that it basically has the same body organs that all chordates do. It has a large and obvious liver. Its lungs are very sort of small and not very well developed, like rest of the terrestrial, well, like mammals and birds. It's going to have a digestive tract with stomach and intestine. It has a urinary bladder. It has kidneys. It has reproductive organs. We have separate sexes in all chordates. So the dissections of the frog and of the shark and of the rat that we have in lab, one of the things to notice is that even though they look fairly different from the outside, they have the same basic body organs on the inside. So they're very similar in those body structures. So reptiles are a fairly large group, and they're going to include things that we call crocodiles and alligators, that there's snakes and lizards and turtles. There's a few other names of kinds of reptiles that we might see, but these are the most common kind. And the tetrapods that are above the amphibians on the tree diagram are going to have the characteristic of having these dermal scales made of the molecule keratin, right? So the reptiles have them on the outside of the body, forming tough, protective, waterproofing coating. In birds, the scales have been modified into feathers, so feathers are made of keratin. In mammals, the scales have been modified into a general reinforcement of the structure of our skin. So our skin isn't totally waterproof, but it's pretty waterproof. It is not as tough as a reptile skin, but it is still reinforced with keratin. Now our hair and our fingernails and toenails are also made of keratin. So this has become the molecule of choice for making these sort of skin-related body structures in the tetrapods. All right, and again, I find that students are fairly familiar with the idea of what reptiles are, so we are going to not talk about them too much. So within the reptiles, there is a clade of the dinosaurs, and within the dinosaurs, there is also a clade of the aves, the birds. So birds are actually the living relatives of the extinct dinosaurs. So whatever few dinosaur-like organisms survived the extinction event in Cretaceous 65 million years ago, they gave rise to the birds. So some people call them feathered dinosaurs. That makes them sound more sort of exciting and cool. I don't think you need to do that to see that they are very exciting and cool. They have a couple of characteristics that I'm going to highlight, although we aren't going to spend very much time on them. They have feathers. They have a 4-chambered heart. They have other modifications of their skeleton for flight, right? So we're going to examine some of those characteristics in the lab today by looking at their skeleton, for example, the modification of their breastbone to allow those extra large pectoral muscles for flight to attach to it, the fact that their skeletons are hollow. They don't have any teeth in their bills, so they have a wide variety of sizes and shapes of the bill depending on what food that they are eating. The other characteristic that birds have that they share with mammals, although they evolved it separately, is endothermy, right? They are going to maintain their body at a near constant temperature. And the reason they can do that is because of the feathers. So we have hair or fur that protects us, or that insulates the body temperature, allows it to stay at a more stable temperature. We expend energy in order to stay at that stable temperature. Mammals do, and birds do too. Mammals, here are some examples of different kinds of mammals. I'm personally quite fond of sloths and bats. Mammals are characterized by having hair or fur, by producing milk. Have a four-chambered heart and are endothermic. We have a range of reproductive characteristics. So we might think about having placental development as being a mammalian characteristic, but we actually have monotremes, which are represented on this slide by platypus. So platypus and echidna are monotremes. They have hair and fur, they produce milk, they have a four-chambered heart and are endothermic, but they do not have placental development. They lay an egg. They lay an amniotic egg. The other interesting thing about their structures is that although they produce milk, they do not have the handy latching onto structure for the babies that we associate with mammals as well, which is a nipple, right? So the babies just have to lick the milk off of this sort of region in on the body where it's being secreted. And those milk glands are actually modified sweat glands. When we look at the marsupial mammals, they have a short placental development. The placenta is formed slightly differently than the eutherian mammals, which have a long placental development, right? So there's still variety in how we go about reproduction, even among the different mammals. So mammals are mostly terrestrial, but we do have ones that have gone back into the marine environment, like this manatee here. We have other marine mammals like dolphins and whales as well. All right, so lots and lots of interesting diversity among mammals. One of the things that we're going to see is that their skeletons and skulls are going to allow us to tell apart these different groups from one another. All right, this slide is a little out of order. So we talked about internal body characteristics and external body characteristics of sharks already. So for now, I'm gonna skip this one. This is for the shark dissection, things you're going to be able to see. And I want to talk a little bit about skeletons and skulls. So when we look at fishes and what kind of skeletal elements they have in their body that allow the evolution of limbs, of the land-going structural supports, and that eventually also led some of the groups to be able to fly. Right, so we'll see that the little fin bones that we see in the skeleton of fish, right, because the fins, they just are these little toothpick-like bones. They don't have a big sturdy structure that fastens to the axial skeleton or the vertebral column. So those had to thicken. They had to develop some different elements. And when we look at a fossil fish, Eusynopteron here in this slide, it shows the development of those bones into kind of just a stubby, thick limb that was or a fin that was supported by these bones. So it could maybe put those fins and push itself up on them, but it really couldn't walk on them. So early in the evolutionary history of tetrapods, these bones formed. They were used for something else. They were not used for walking on land, but because they existed already, as they were modified and to get larger, it allowed the earliest of terrestrial or land-going, somewhat land-going chordates to take advantage of the resources in the terrestrial environment, whether to feed or whether to hide from predators. We don't know exactly what motivated them to crawl out of land, but probably some combination of those things. So in all of the tetrapods, we see this basic body ancestry in the bones that we see in their limb, right? Whether we're looking at a frog or a rabbit or a lizard or a bird or a human, we see that upper arm bone that's sort of thicker and one solid bone. We see the radia and the ulnus, which are the two smaller bones that we see next to one another in the lower limb. And the reason that is important is that the way that they're fastened together allows our arm to twist, the hand to twist. We see groupings of these smaller bones, the metacarpals and the phalanges that make up the fingers, right? So all of our tetrapod bodies are based on these same little bone elements. They may be modified a great deal, but they're still recognizable that they came from an ancestor with the same characteristics. So whatever the limbs we end up having on our tetrapods are, we see that those limb bones represent a shared ancestry. All right, when we look at diversity among the amniotes, right, another way to talk about the tetrapods is to refer to that amnion and say they're the amniotic chordates. We see a distinction between reptiles and mammals on our tree diagram. And we're going to focus on, for a moment, on the characteristics of the skull and skeleton that allow it to make this distinction. Right, so we have reptiles over here, and the word I want you to clue in on here is diapsids. And so most reptiles are diapsids, and mammals are synapsids. And so the characteristics of the skull that separate them into these two groups, we're going to look at what those are in mammals that makes them synapsids. All right, so while I have this slide on the screen, we have the names of the taxonomic groups for reptiles, squamates, which includes lizards and snakes, tuataras, which are lizard-like but have some unique characteristics. We have the extinct dinosaurs, and that we can see from this one that in the dinosaur clade, the only living taxonomic grouping is the birds. Crocodilians of the living taxa are most closely related to birds. And then turtles are over there on the side. We don't completely understand the relationship of turtles to the other reptile taxa. All right, so we're going to come back to this idea that mammals are synapsids and what is a characteristic that just defines them as a synapsid. All right. That has to do with how the jaw joint is formed. It has to do with the holes that they have in their head. So this is the eye socket. That's where the eyeball goes. And this other hole here is called the temporal fenestra. That's a hole on the side of the head. Synapsids have this hole. And the diapsids have two holes on the side of their head. And what does that hole allow you to do? The hole allows a muscle to go over the bone at the bottom of it and fasten onto the lower jaw. And so the way that the jaw is articulated or fitted together and allowed to move in the synapsids allows chewing. All right, so it doesn't seem like that big of a deal, but when you're trying to get the most energy that you can from your food, chewing it up is a way to be able to get more energy out of your food. So biting and chewing are very important in evolutionary history. So the synapsid characteristic, mammals have this structure called the temporal fenestra that allows them to chew their food thoroughly. All right. Difference between reptiles and mammals also has to do with some tiny little bones in the inner ear. And so these relate to how hearing is accomplished. Reptiles only have a bone called the stapes, right? Mammals have not only the stapes, but they have a bone called the incus and a bone called the malleus, the hammer and the anvil. And so those make a little bit more complicated connection inside the ear and relate to how hearing is done. Right? These are tiny little bones. They're hard to find in the skull, but anatomists have noticed those for a long time. All right, so a couple different changes in the skull that separate the synapsids from the reptiles or diapsids. Another thing that is true in mammals that allows a lot of evolutionary diversity is that there's 4 kinds of teeth. And ancestral version of all of the teeth in mammals, remember, because now we can bite and chew because of the way the jaw fits together, that having different kinds of teeth allows you to take in and process a wider variety of foods. And then we see over evolutionary time that the sizes and shapes of each of these kinds of teeth has been modified to suit different kinds of diets better. So when we look at structure and function, right, we have these 4 kinds of teeth. Incisors, which are the 4 little flat ones in the front of our mouth. We use them for biting. Canines are a big pointy tooth that we find mostly enlarged in carnivores that allow biting and holding on to a prey. Premolars are sort of the next set of teeth, and in carnivores, they're sharp. They aren't sharp in our mouths, but they're a very versatile kind of tooth that's widely changed in its structure when we look at animals with different kinds of diets. And then the molars in the back, so the premolars and then the molars. The molars are the big flat tooth. They're flat, but they have some little pointy structures sticking up. And then when we grind them together with the teeth on top, then we can accomplish a lot of mechanical digestion for food. So when we look at these different kinds of teeth and we look at them in different skulls of organisms, we can tell what diet that organism has from the way those teeth are shaped. So we have modification of the premolars. If they're sharp for slicing, then we're looking, and the canines are big, we're looking at a carnivore. If there's a reduction in the canines, sometimes they aren't present at all, and our premolars look like molars, and our molars are really big, then we have an herbivore. If we have kind of in-between teeth of all of these kinds, then we're looking at the skull of an omnivore. So humans are omnivores. We just basically have biting and chewing teeth. We aren't really specialized for one kind of diet. We eat all kinds of things. All right, so when we're looking at evidence from fossils, we can learn a lot, not only about the structure of the skulls from looking at them, but the function, because the skulls are modified, the teeth are modified, the skeleton is modified based on the kind of diet and the kind of ecological role that that organism has. All right, our last slide here is just reminding me to say that in your rat dissection, you are also going to get to see the same basic body structures that humans have and that all of the chordates have. So although we have a wide range of bodies in the chordates, from those weird little tunicate animals up to humans, that we have an extraordinary amount of shared characteristics, ranging from the internal organs to the molecules that make up our skin surface, to the elements of our skeleton, repeated body segmentation, and when we are going through early development, those 4 chordate characteristics. All right, enjoy your lab activities today.